Okay, so this chapter, chapter 11 in Campbell's, kind of represents a transition point in the class. Obviously, we've been stuck in metabolism for what seems like forever. And the last few chapters, studying how the cell gets energy to do the work it needs to do. But not all pathways have to be metabolic pathways. So it's kind of nice because this next section, we're going to look at a few pathways that are involved in signaling. And this takes us to the next section where we're going to look at DNA structure, and DNA function, and gene expression. And as it turns out, most of the time the cell needs to respond to external stimuli. It's going to rely on the DNA for instructions to do that, basically. So the DNA, you guys already know DNA and RNA are the information molecules. So they have the information that's required for the cell to basically do everything. So our chapter today kind of looks at, kind of bridges that. So we see where the cell gets all its energy from. It uses that energy to do things, endergonic processes, which if we're talking about making proteins or making DNA, those are all going to be endergonic. So that's they're going to reach back and get the ATP from the metabolic pathways to use to make things. Let's see, what else? Oh, so signaling pathways help the cell to respond to stimuli. If you go back to the first chapter in Campbell's, it talks about some of the characteristics of life. And one of those is the ability to adapt or to sense stimuli and react to it or change something. And so signaling pathways are really important to get that done. It's also, as an example, as we'll see in here, of regulation. Remember I told you several ways that the cell can regulate or control pathways. One was gene expression, whether you make the protein or not. The second one was modification, right? So you put a phosphate group or something on a protein, you can turn it on or turn it off, depending on what the protein is. We're going to see a lot of examples in these pathways of proteins acting on other proteins, and that activates those proteins. So then they go and do something else. Okay? So that's kind of what this chapter provides. We're not going to have to memorize any of the pathways, fortunately, but I just want you to kind of take a step back and look at, appreciate what these pathways do, the complexity, and why they're so important. Okay? So that's our question for today: is how do cells respond to stimuli? And it doesn't matter if they're prokaryotic or eukaryotic. So if we're talking about prokaryotes, they're single-celled organisms living by themselves in a big cold world, right? They need to be able to respond to things that are changing in their environment. They do it through pathways like this. Eukaryotes, what about us? Do our cells communicate to each other?
Yes.
That side of the classroom said yes. What about this side? You guys agree? Yeah, absolutely, absolutely. And it's when they stop communicating with each other when we have issues, or they start ignoring messages from other cells. Okay? Yeah, eukaryotes is very, very important. It's a lot of crosstalk, if you will, between organs and cells. So, some examples. Here are some examples for single cells. Bacteria. They can interact with each other and actually change behavior. So there's examples of cells signaling to other types of cells that then change them into a more aggressive type, and maybe they can cause disease, or they can secrete chemicals or something that can hurt the host. So there's examples of this where it's almost like an army or something sending messages, and then they get recruits and they go on the attack. Another one is biofilms. Have you ever heard of a biofilm? Like this film of bacteria, really dangerous in medical settings sometimes. They tend to inhabit the, what do they call it, the tubes, the feeding tubes or the breathing tubes that you put into people. So inside there, you can actually get coated with bacteria. And what the bacteria do is they send out signals to recruit other bacteria, and they start to form kind of a community. And the community actually provides protection from the outside. Even antibiotics sometimes have hard times getting into biofilms to kill off the bacteria. Anybody know another example of biofilm in our bodies? Skin, I'm not sure there's actually a film. There's certainly bacteria. Close. Everybody brush your teeth this morning? What happens when you don't brush your teeth? Right now, psychologists say we all get our tongues, right? And check our teeth to see how smooth they are. We just talked about it. Right. If you don't brush your teeth, you get that white, nasty, filmy stuff on your teeth. That's a biofilm. So bacteria that have formed this filmy substance, and it's pretty resistant. I mean, to get it off, you've got to use toothpaste, which has grit in it, right? It has kind of a sand compound in there to brush that stuff off. And if you don't, then they can start to cause cavities, right? So biofilms are kind of an interesting relationship between different bacteria. It's kind of cool. Let's see what else. There's a lot of studies into quorum sensing. So bacteria actually communicate with each other to prevent overcrowding. So that would make them run out of resources faster, so they can actually slow down their growth by communication. What about something more simple? What about just changing what you're eating, what you're metabolizing? Would it be good to sense this for a bacteria? Sure, I mean, if you're growing, this is bacterial density on one side. As you grow, you're using glucose, usually is the first common or preferred sugar source. What happens as they run out of glucose? You kind of hit a plateau, yeah? So what are they doing in that little plateau? Just giving up and rolling over? No, what do they do? Adapt. They can try to sense, maybe there's other sugars they can use. Maybe there's other sources of energy. So this adaptation here, this little chase, I don't know what to call it, it's a plateau in growth. They're not dead, but they're changing the way they're metabolizing to try to find something else they can use. So maybe you have something else like lactose in there. So now they can start breaking down the lactose into glucose and then use that to grow some more. So it's kind of a very simple, we can use pathways to switch. The way, that's a characteristic basically, is you're switching what kind of sugars you're using, if you can. So pathways, very, very important in sensing what's going on outside of the cell and then responding to that. You're kind of internalizing the message. Okay, we're going to kind of focus on eukaryotes from here on out. We haven't really talked about cell characteristics that much, but there are some cell-cell characteristics that can help cells to communicate with each other. So for example, gap junctions are these little gaps in the plasma membrane between two cells. You can find this in heart muscle cells. They can communicate with each other using small ions, for example. Plants also have the same kind of idea. It's called plasmodesmata. But most other cells, they're going to have proteins on the outside of the cell, on the plasma membrane, that are going to be required as part of this communication. So we want you to start thinking about these proteins. They're almost like enzymes in the sense that they're going to be able to bind to different signals and then respond to the signal in one way or another. So here, structure, having the correct protein is going to be important for responding to signals. Okay, let's talk about the types, just general features of different signaling. I want you to know these by memory, not for this exam, right? So this is, after this exam, then you can make your flashcards with a list of terms and learn them all, okay? So first one is endocrine signaling. These are just general ways that cells can signal to each other. So here, what you can see in this picture is an endocrine cell or any kind of hormone secreting cell. It's going to release those hormones, which are typically small molecules. So, I don't know, insulin, adrenaline, these kind of things are small molecules that can float around the body. And then those signals are basically, I mean, why do you use the bloodstream here? Why is that convenient? Yeah, it goes throughout the entire body, right? So you're basically flooding the entire body with this message, whatever it is. And they tend to be a little longer lived, or longer lived, however you say it. So they stay stable in the bloodstream for longer periods of time. But does every cell respond to those signals? No. So there's only going to be certain target cells that are going to have a specific receptor, typically a protein, that's going to be specific for this signal. So just like an enzyme is specific for its what? Substrate. A receptor is going to be specific for its signal, whatever it is. So if it's insulin, we're going to have an insulin receptor. If it's a sugar even, sometimes we have a sugar receptor. If it's a growth hormone, then you're going to have a particular receptor for those growth hormones, because there's more than one. Okay. Let's see. Try to come up with examples for these. The one I always use for endocrine. Oh, and by the way, think end, like ends of the earth. So long distance. Talking about long distance signaling. Endocrine is long distance. I always think of puberty. Yeah? So age, I don't know what it is nowadays, 10, 11, 12, seems like it's getting younger all the time. The pituitary starts dumping hormones into the bloodstream, and then what happens? All hell breaks loose, yeah? Well, males and females, it seems like. Yeah, and what are those hormones doing? When they're going to the sex organs, they're going out, whatever, and you start to sexual maturity, and those things start to produce their own hormones. So a lot of hormones racing around, right? In teenagers. The link groups them with teenagers. Sorry. No offense. Okay, good. Okay. So I think endocrine, long distance signaling. So the pituitary is dumping it into the bloodstream. It goes throughout the body, but then only certain target cells will respond. Does that make sense? Why don't you think about this idea that the target cells have to have the correct receptor for the signal in order to even start the process. Yeah.
How does the cell tell whether a hormone is in the bloodstream or not? How does it decide it's going to extract it from the bloodstream?
It doesn't extract it, it diffuses, right? Through the capillaries. So they're usually small molecules that diffuse. We'll come back to that, but sometimes it depends on whether they're hydrophobic or hydrophilic. So, but this just shows it popping out of the bloodstream, but it would probably be through the capillaries and others, and the lymph system as well. Yeah.
Does something need to trigger an endocrine cell to release its hormones?
Yeah. So these cells that are secreting things, they don't do it on their own. It's usually some signal above that. Yeah. So this one would probably be responding to a signal from something above. Yeah, there is kind of a hierarchy. It's kind of interesting. Okay, paracrine is another example. This one I think of as short distance. Instead of long distance, we're talking about short distances. So these signals then are a little less long-lived. They're a little more unstable, so they don't last very long. So they're only going to target cells that are kind of in the vicinity of the secreting cell, whatever it is. So a good example is platelets, platelet activation. You guys know what platelets are? What do platelets do? Clots, right? So you get a cut, you want the platelets right around that area to be activated. Why don't you want that to be endocrine signaling? Yeah, because blood clots elsewhere, like in the heart or lungs or leg, will kill you. Yeah? Well, maybe not in the leg unless they go to the heart, right? So, yeah, you only want blood clotting, that kind of activation in the general area that you want it to be. So that one, you don't want to have long distance. And some people have issues, right? If you're on planes a lot, or if you have some genetic mutations or something, sometimes your platelets activate when they're not supposed to. So. Okay, this one down here in the corner is neuronal. It's pretty self-explanatory, right? What kind of cells do neuronal signaling? Neurons, okay. So in physiology, you'll learn how the neuron is activated. It sends the signal down the long axon, and some axons can be really long, right? What is it? Sciatic is a meter long or something like that. So you'll learn all that. It involves membrane transport and pumps and shuttles. So you'll see all that again when you do that. But then the action potential it releases a bunch of neurotransmitters, which then signal to the target cell. And the target cell could be another neuron, or it could be a muscle cell or something else. Okay. So neuronal signaling is just for neurons. You won't really talk about that much. What about contact dependent? What do you need? What is it dependent on? Contact. So this is for cells that are right next to each other. A classic example, if you look at neuron development in the brain. When it's first developing, not all brain cells become neurons. They're differentiating. Some are actually told to die off. And it's done through this kind of cell-cell communication right next to each other. Or it's told to do something else, maybe become a Schwann cell or something else that helps the brain. So contact dependent is right next to each other, right? Which is a little different than paracrine. So we have neuronal, contact dependent, and then I'm going to add one more. It's kind of interesting. Autocrine signaling. Anybody heard of this one? Auto, what do you think auto means? Autobiography? So this one's kind of weird. It's a cell that will signal to itself. Send out a signal to itself. Good example is in the immune system. When you look at B and T cell development, B cells will actually communicate to themselves and tell themselves to divide. So you get what's called a clonal expansion. So tons of cells being made through that process. You guys ever have swollen lymph nodes? Because they're packed with expanding cells, B and T cells. So getting ready to fight an infection. So autocrine is a cell signaling to itself, believe it or not. Okay, so put them to flashcards, write a sentence to describe them so you know the terms for exam 3, not exam 2. Okay? Any questions on that? General types of signaling. What else? Did I already mention that our cells pretty much rely on signals to do everything? It's like the military inside our bodies. So whether they need to grow, divide, or even die, they always rely on signals to do that. When they stop listening to signals, that's kind of where you get into cancer cells. Cancer cells kind of do their own thing. They don't listen to anybody else. It's usually not very good for the host. So our cells are pretty closely regulated when everything is working properly. What kind of signals are there? Well, if they're small hydrophobic nonpolar signals, can those diffuse right through the plasma membrane? Yes. So when we see things like those, like cortisol is a good example, or nitric oxide, those things are pretty small, but they're still going to have a receptor. They're still going to have a protein that's on the inside of the cell that they're going to bind to start the signaling process. What about large polar compounds, like insulin? Can they get through the plasma membrane? So where are their receptors going to be? In the plasma membrane. In the plasma membrane. So, yeah, on the outside of the cell. Okay. And they're going to be able to bind things there. Okay. What kind of receptors are there? So this one, don't memorize, okay? Just take a step back, and I want you to appreciate the complexity of what these things are. There are channels. What are channel-linked receptors? What are channels? Do you remember? Transport? Yeah, they're facilitators, facilitated transport. So these channels, when they bind the signal, they open, and then you get a rush of ions through it, and then they can close again when the signal comes off. Okay? So they open and close depending on the signal. Some are called enzyme-linked receptors. So on one side, you bind the signal, whatever it is, that chain has a conformational change in the enzyme, the protein, and now on this side you have an enzyme domain that will then generate some sort of a product, and then that product will go on and complete the pathway, basically. So kind of interesting. Two different parts. This obviously would be transmembrane proteins, wouldn't they? They'd have to be across the membrane. Another example of GPCRs, G protein coupled receptors. Really important in humans. We have, I don't know, over 200 different kinds. And they're involved in sight and smell and taste, so they're pretty important. These guys bind to a signal, and then they activate what's called the G protein, and then the G protein will go on and activate other proteins to start the signaling process. And then this one just shows an intracellular one where it binds and then changes conformation when you start it. So, cool, right? Structure and function, pretty important here. Now you got to have the right structures for these receptors in order for them to function properly. Right? If they don't have the right structure, they won't be functioning properly. Okay, let's look at the steps of reception, or the steps of signaling. First one is reception. Well, that's kind of important, isn't it? Do you want the pathway to just start whenever it wants to? No. So a pathway to turn on should always start with binding of the signal. Binding of the signal. So the first step is reception. And in order to have that, you have to have the signal and the receptor, don't you? So not all cells will have that receptor. And so those cells will or will not respond to that signal, whatever it is. So reception requires both. You have to have the signal and the correct receptor coming together to start the process. If you're missing one or the other, you don't want to start it. That's not a good thing to do. Okay? So reception is the first one. Reception. Pretty straightforward. Just think of it like that enzyme-substrate relationship. You have to have two to tango, right? When it comes to metabolism. I just came up with that one. Is that pretty good? I'll have to keep using that. That's good. Okay, the next step is called transduction. Transduction. I always think of this as kind of a relay race. You guys done one of those? With the baton or beanbag or something? So here, what we're going to see is that there's a pathway of proteins that are going to be interacting with each other, and they're going to be passing down this message, whatever it is. Notice they're not passing the signal molecule, are they? If you look at this picture, the signal molecule is still outside the cell. So these proteins are going to interact by activating each other. That's how they're going to pass the information from one protein to another. I kind of think of it as a relay race or some sort of a cascading effect, how they're going to act on each other. I'll show you an example here in a second. So I think of kind of like a relay race. But here what we're going to see is that the proteins interact and activate other proteins, basically. And they're doing this to relay a message. To relay a message, okay? Let me give you an example. And this might be a little scary, so hold on to somebody next to you. Okay? It's okay. So this is a protein cascade right here. So you see the signaling molecule there, there's the reception going on. And then what we see here is there's several inactive proteins just kind of floating around. Remember, proteins can be in an inactive state. They're still there, but they're not actively catalyzing the reaction. Here what we see is once this pathway is activated, then we start activating these kinases. Kinases are proteins that transfer phosphate molecules from one place to another, either between proteins or a protein and a nucleic acid or whatever. So this one is activated, and now all of a sudden it activates this one by putting a phosphate group on it, and this one activates another one by putting a phosphate group on it. See, this is an example of how we're modifying these to activate them. So do you remember that? When we talked about the steps of regulating a protein? I mentioned it already. Gene expression. This is modification, number 2. Inhibitors was number 3. Remember that list? So here, we're modifying proteins to activate them. And that's how we're sending the message. That's how we're relaying the message. Kind of like a cascade. So we activate one protein, we activate another, we activate another. And it depends on the pathway, but you could have 10 or 20 different proteins here. Or just a couple. So it really depends on whatever pathway you're looking at. Kind of interesting. Just take a step back and go, wow, that's kind of cool. It is a pathway, isn't it? A bunch of enzymes working together, but they're not really metabolizing anything, but they are interacting and sending, kind of transducing a message. Yeah? Okay. You're doing great. Hang in there. Come on. Maybe it's just not exciting. Okay. So that's transduction. What do you think the next step is? Response. So obviously, you want the response to be related to whatever the signal was, okay? And that's what we're going to see, is that if you get a signal to divide, then the response from this pathway, from this signal, will be starting all the cell division cycles and starting those proteins doing whatever they're supposed to be doing, okay? So the cell response is going to be related to whatever the signal is. Number 3 is the response. And if you want the response to be whatever you're supposed to do. So if it's a change in cell characteristics, like a white blood cell or something can get a message and then change the way it moves around, that's one response. Or I mentioned cell division, you're going to have cell division starting. Or if it's to secrete a different message, right? Then it'll make that message and secrete whatever the response. This typically involves the nucleus. Not all the time, but most of the time, I would say. It involves the nucleus because what's in the nucleus again? The DNA, isn't it? Right? So in the DNA is all the instructions and everything the cell needs in order to make whatever response that it's supposed to make. Okay? So the DNA is the source of all the information. So when we look at some of these pathways, we'll see they typically go through the DNA. 3 stages. Any questions on the 3 stages? Yes?
Can you repeat like the wording you used for when you said like the end would be like kind of like similar to the beginning signal? What was like the phrase / wording that you used there?
I don't know. I was just trying to say that whatever response you want, you want it to be related to whatever the signal was telling you to do. Is that that's what I was trying to say. Was that if the signal says grow, then you want the cell to grow. So the cell response will be changes in gene expression, what we're going to look at in the next few chapters. Changes in gene expression that will bring about that response, whatever it is. So if it's cell division, then we start making the proteins that the cell needs to divide. Yeah. Did that help?
We're good,
Okay.
thank you.
That's fine. Sometimes it's good to try to restate it, I don't know, a couple different ways. Okay, so there are some examples. Let's see here. So here's one where you're binding to a receptor, but it goes right into the nucleus, and then you get some sort of a change. We already looked at that one. Here's an example. So two receptors coming together to activate a pathway, and then you notice it comes into the nucleus here. Okay? We'll learn this term called transcription factors. These are proteins that are going to be involved in the pathway that are going to interact with the DNA to start transcription. So when we look at transcription, we'll come back and mention these pathways, okay? Here's another example. This is the RAS-RAF pathway. Very common, this pathway is very commonly mutated in cancer cell lines. You guys know what the definition of cancer is? Yeah, abnormal growth or uncontrolled cell division. Uncontrolled cell division. All cancers have that in common, where the cells just divide no matter what. And that involves these pathways which are involved in cell division. And I think this reiterates the point. You want to be able to turn on these pathways and divide, but you also have to be able to turn them off as well. So either to deactivate the proteins, get rid of the signal, whatever way you can do it, you want to be able to stop them as well. Very, very important to control these pathways, because that's when cells start to do whatever they want to, is when the pathways just turn on for no reason. And that's not a good thing. But notice here in the end here, ERK is a transcription factor that's going to come into the nucleus and then start changing the gene expression for cells that are required in growth. So that's what I'm trying to convince you. This is a bridge. Here we're looking at a pathway that's changing gene expression, and now we're going to get into gene expression in the next few chapters. So that's why I think it's kind of cool. Oh, you guys heard of fight or flight? Now, some pathways are really slow, like cell division may take an hour and a half or so to complete in humans. Fight or flight pathways, you don't want those to be slow, do you? Yeah, if you're running from a, I don't know, cheetah or something that's going to kill you, you want your cells to respond really, really quickly, don't you? So here's an example of cortisol. Cortisol signal is very, very fast. And that's because it binds to its receptor and goes straight into the nucleus. And this has lots of different effects, but one effect that it has is that your liver will dump glucose into your bloodstream. Why is that a good thing? Yeah, energy. Because if you're running for your life, you need ATP. Right? And you don't want to, hopefully you're not going to ferment, okay? So, yeah, these things, and it has other effects as well, right? It dilates your eyes or something, I forget. There's numerous effects, but some pathways are going to be really, really fast. I think I have a picture here, yeah. Where it just goes very quickly in seconds. Others are going to be a lot longer. So it really just depends on the pathway, which way it's going to take. So that's cool. And there's other examples like adrenaline does kind of the same thing. So, okay, we talked. Yeah, that just shows different ways the pathways can respond. Yes, just like metabolic pathways, signaling pathways don't work in a vacuum. So if you get into this kind of research, you're going to see that these kind of signals, there's a lot of crosstalk, I use that term again, but pathways interacting with each other. And usually you have to have multiple pathways being activated in order to get a response or get the correct response. So it's very, very convoluted, and I've done some work on some of these pathways. It can be hard. Certainly I don't memorize that, whatever you do. Okay, so what's the point? Well, like I said, we're making a transition. We're going to see, so we see now that some pathways are not metabolic pathways, but they're involved in other processes. Now, starting on, what, Friday, right? Because we don't have class on Wednesday. Starting on Friday, we're going to look at DNA structure and then get into gene expression later next week. But we're going to see that the way the cells change their behavior or change their characteristics comes through these pathways being activated. And the pathways are going to interact with the DNA. We're not going to change the DNA, but we're going to change the way we use the DNA, and that's going to change the cell. Okay? So this chapter kind of represents a transition into the next section. DNA is always fun, right? Much more fun than metabolism. So, okay, any questions?